Hey, in this video, we are going to be talking about the decarboxylation steps that are part of cellular respiration. So in a different video, I went over glycolysis, where we got glucose and we broke it down into pyruvate, and we got a little bit of energy out of it. Now, that is not the end of the story when it comes to breaking down sugars. So the concept of breaking down sugars through cellular respiration is shown here. And after glycolysis, we go through another few steps, which I call the decarboxylation steps. And so that is what we're going to be discussing in this video. As you can see in the decarboxylation steps, we're going to have two things, pyruvate processing and the citric acid cycle. This is, by the way, happening in the matrix of mitochondria if you are a eukaryotic cell. If we're talking about prokaryotic cells like bacteria, this is happening in the cytoplasm. And during this process of decarboxylation, we're going to be producing even more energy. So the first part in this decarboxylation stage is pyruvate processing. So remember, pyruvate was the end product that we got in glycolysis. And what we're going to do is decarboxylate it. Now, what this means is we're going to remove a carbon. Remember that during glycolysis, we had 6 carbons in the beginning for glucose. And at the end, we had 2 molecules of pyruvate that both had 3 carbons. So we didn't lose any carbons. We had 6 carbons, and then we got 3 plus 3 at the end. But now we're finally going to start getting rid of these carbons through decarboxylation, and basically the carbon is going to be removed and released as carbon dioxide. And during this process, we're also going to be oxidizing the pyruvate. Remember that whenever we oxidize a carbon molecule, we're going to be extracting energy out of it. So we're going to decarboxylate and release the CO2. And we're going to extract some electrons out of this molecule, and those electrons are going to be added to NAD plus to produce NADH. Remember that NADH is the charged version of NAD plus. So that's going to happen. And then the last final step in this process is we're going to add a coenzyme A molecule to the final product. And so the final product that we get is going to be acetyl, so an acetyl group, and then a CoA. And that's the end. And remember that the acetyl-CoA has 2 carbons, which makes sense. We started with 3 carbons, and then we end up with only 2 carbons because one of them we released as CO2. And that is the pyruvate processing portion of cellular respiration. And again, this is happening in the mitochondria or in the cytoplasm if you are a prokaryotic cell. Now, this acetyl-CoA is going to go through further processing, and this is called the citric acid cycle. So there's another name for it, the tricarboxylic acid cycle. There's also the Krebs cycle, but Krebs kind of got canceled, so we stopped using that name. But anyway, it is this process where we're going to get this acetyl-CoA, and we're going to basically decarboxylate it even more, oxidize it even more. So we're getting rid of more carbons. We are extracting more energy out of it. So what's going to happen is it's going to be decarboxylated 2 more times. So we're going to release 2 more CO2 molecules. Now that is all of the carbons, right? So acetyl-CoA only has 2 carbons. If we release 2 CO2s, that's it. There's no more carbons left. So we have basically fully gotten rid of all of the carbons. And then again, during this whole process, we are oxidizing the molecule. So we're going to make 3 NADHs, 1 FADH2, and 1 ATP, or GTP, by the way. So GTP is like the G version of ATP, if we're thinking of the nucleotides. So instead of the A nucleotide, it's a G nucleotide. But same thing, we're going to be making an ATP. By the way, when we make this ATP during the citric acid cycle, it is through substrate level phosphorylation, the same kind of substrate level phosphorylation that was done in glycolysis. And that is pretty much it. So now the acetyl-CoA is completely, broken down into its carbons and completely oxidized. So we've kind of extracted as much energy out of the molecules as we can. Oh, by the way, the CoA just gets removed at the beginning. So we add it, and then we just remove it. So it was only there for a temporary moment. And here's the citric acid cycle in a little bit more detail. Again, TCA cycle, citric acid cycle, same thing. So again, we are starting with acetyl-CoA. Remember that has 2 carbons. it is going to enter the citric acid cycle by combining with this other molecule called oxaloacetate, which has 4 carbons. So 4 plus 2 is 6. We're going to be making citrate, which has 6 carbons. And somewhere along the lines, the CoA was removed. Don't worry about that too much. Now we're going to decarboxylate it and oxidize it. So we're removing a carbon and we're also making some NADH. And we're going to make this 5-carbon molecule called alpha-ketoglutarate, AKG for short. And then we decarboxylate again, and a bunch of other oxidation steps happen. Again, I'm kind of hand waving here. I don't want to go into too much detail, but stuff happens, and we lose another carbon. So now we're at succinate, which is, again, we're now down to 4 carbons, right? So we went from 4 plus 2, which is 6, down to 5, down to back to 4, basically. Okay, and then this succinate gets oxidized a bunch more times, and we eventually get back to oxaloacetate. And so this is why we call it a cycle, because we do go back to oxaloacetate, and then it basically just keeps going over and over again. But again, the key steps that you should know is that there's a decarboxylation step for the citrate, and then there's another decarboxylation step that eventually makes it into succinate. Again, there's a lot of other steps that I'm skipping. So these arrows here have gaps. There's other molecules that are produced in between these steps, but I just don't want to go into that much detail in this video. And so these are kind of the main steps that are happening in the TCA cycle. So now what? Well, we have fully decarboxylated our glucose, right? So in glycolysis, we had 6 carbons, which we split into 3 and 3. And then during the decarboxylation steps, we basically release all of it as CO2. So we're going to end up with no carbons left in the molecule itself. Now, in another video, we're going to go over the electron transport chain because it's not over yet. All of these molecules that we produced that are electron carriers are going to go into the electron transport chain to generate even more ATP. So I will see you in that next video, and hopefully this was helpful.